The alternative complement protein system is reliant not on antibodies, but it's independent of antibodies. The largest part of complement proteins is probably complement 3. Now, complement 3 is produced predominantly in the liver. It's also accessible in other tissues throughout the body, but it's predominantly produced in the liver. Now, C3 is developed into C3b during processes of activation of the complement system. And what this does, it enables our bodies to be able to recognize hydroxyl and amine groups on bacterial or other pathogenic or antigen surfaces. What this does, it pretty much attaches itself onto these hydroxyl amine groups, and it starts a whole series, a whole cascade. In the instance where there isn't any attachment, it becomes neutralized by attaching itself to water, instead of creating a host of unwanted immunological response inflammation and so on. However, when it does attach to it, what happens is there's another complement protein B that attaches to the C3b molecule itself, and this attracts another complement D protein. In essence, what this does, it actually creates an enzyme called convertase, which is basically C3b, lowercase, B uppercase, lowercase b. Again, so it's complement 3b, upper b, lower b, which we call convertase. Now, this enzyme basically creates more and more of its own convertase by splicing and clipping C3b molecules. And in essence, what this does over time, it clips a C5 molecule, okay, into two different pieces. And when we clip the C5 molecule into two different pieces, now we're getting started, okay, because this creates a major attack complex. So the C5, one of the parts of the C5 complement protein that gets split combines with C6, C7, C8, and C9. Now, C9 is very, very important. C9 creates these little channels, and these are creating holes in the bacteria, punching a hole through it. And this whole entire major attack complex can now kill unwanted entities in the body by creating huge punch holes into it. However, it's very, very important that when we do activate the system, that we don't destroy our human cells. And how we go about this is by three big players. One is MCP. MCPs basically are proteins that stop the clipping of 3CB protein filaments into becoming active convertase into 3CBBB. In addition to that, there's another player called DAF, and it's called the decay accelerating factor. Okay, so what this does, and it inhibits the convertase that's already there from being able to attach onto more 3CB protocols and creating that whole punch sequence of 3C5 protein molecules being splitting up and creating that major attack complex. So this is DAF, or decay accelerating factor. Okay, very, very important we have this in our human cells to prevent them from being destroyed. And the last one is CD59. This is also called protectin. And what this does, it stops C9 protein, okay, within that whole complex from punching those holes, right, from creating those channels in the bacteria or the bacteria in our human cells, so that we don't actually start to decay our human cells at a rapid rate, which happens in autoimmune diseases. So for those of you who are battling or know of people that are battling with autoimmune compromise, have them reach out to us. Let's see what we can do within that complement protein sequence and try to augment the response. When we actually do that, the best is by looking into the liver. Just to give you a little bit of a FYI there, look into the health of the liver and how effective and efficient it is in producing these complement proteins, as well as these inhibitory factors to their function and into their activity. For me, Justin McGuire, stay healthy and stay optimized. Take care.